Okay, well, it's a real pleasure to meet with all of you, and thank you for your patience with the technical difficulties. My understanding is that all of you are in high school, is that correct? Or are some people already in college or beyond? My understanding is all high school. I'm going to ask that we do this in an interactive way today. And so the way I'm going to set that up is I'd like to ask for a few volunteers before I even get started to interrupt me and ask a question. Can 2 or 3 people volunteer? Just you can react with a hand up or something, that at some point during the next 45 minutes, you will interrupt me with a question. And my hope is that if we assign the few people to do that, then everybody else will also feel comfortable to jump in with a question throughout. Do we have volunteers in case no one else does? While I figure out how to share my screen here. Okay. All right. So I'm going to talk to you guys about the microbiome, which is all the bacteria that live inside and on our body. And I like this comic that I found. The Human Microbiome Project says that the human body has 100 trillion microscopic life forms living in it. And then there's a response here from one of them. You call this living? This always reminds me, for those of you who are traveling to New York for your summer projects, this always reminds me of living in New York City, or at least being on the subway during rush hour. All right. So bacteria. Bacteria are single-cell organisms, single-cell life forms. And for most of human history, no one ever knew they existed because they're invisible to our eyes. But in the 1600s, this guy, Antony van Leeuwenhoek, maybe I'm mispronouncing it, Leeuwenhoek, who was a Dutch guy whose family sold carpets. And because they sold carpets, they had an interest in examining the fibers of the carpets to be able to tell if the carpet is of really high quality or really poor quality. And so he got interested in lenses and magnifying glasses. And he became quite adept at making lenses that were better than everybody else around was making. So he could get better and closer looks at these fibers on the rugs that his family was in the business of selling. And when he put living things, water, under this lens that he developed, he discovered little teeny tiny things wiggling around. And he called them animalcules, meaning tiny animals. But this was the first time that humans ever saw bacteria. Fast forward a few hundred years, we now know that bacteria are absolutely everywhere. And we often in our day-to-day life think of bacteria as something that causes disease. And indeed, many of them do cause disease. But many more bacteria, in fact, most of the ones that live in the world, don't interact with humans in any way, shape, or form. They live out in the soil, or in the ocean, or on surfaces, or on other animals, and most of them don't interact with us in any way. But some of them do interact with us in ways that are beneficial, mutually beneficial, where we help them out and they help us out. And it's this last category that is the subject of my research and what I hope to share with you today. By way of orientation, let me switch to this very cool website called Evogenau, which is this wonderful tree of life that I encourage you to poke around with. This is a tree that summarizes all living organisms, and it also gives the time scale of how they all evolved. And the idea is that all life forms evolved from an early common ancestor. And so that common ancestor, we don't know anything about it really, but it's theorized to be at this point. And then all the different life forms have, over the course of evolution, branched off from it. For example, all the humans here start at this end. We have humans here, and some of our closest neighbors, the chimpanzees and gorillas, and many other mammals. And then we have birds.
You want to just show it? Oh wait, I don't have headphones.
Is there a question?
No, my jacket is either on. Okay.
Okay.
Yeah, I'm gonna turn on subtitles.
Try to mute your microphone.
I'm just saying it. It's like she's
Anyway,
probably taking attendance.
See and the insects and the crabs, crayfish, and then we get into the plants, and then everything we've talked about up until now are eukaryotes, which are organisms that have cells which have a nuclei in the middle. But even more primitive than that are cells that don't even have a nucleus, and those are things like the protists, and then these two large categories, one called archaea and one called bacteria. The nice thing about this website is that you can click on an organism and it'll trace back from humans to whatever organism you've clicked on and tell you how closely related we are to that organism. So the common ancestor of bacteria and humans is our 706th billionth great-grandparent. And bacteria are our 706th billionth cousin, 1.4 trillion times removed. Whereas if you click on another organism, like let's say cats, maybe some of you have pet cats, to find a, so here it links up cats to humans. Anyway, so you can have fun with this if you're interested in biology in general, but that's just to give you an idea about how bacteria are very distant from us and yet also share a common ancestor with us. And we share some common features, like we both have DNA in that encodes our genetic material. We both use proteins, we and bacteria. We both like to eat food like carbohydrates, but slightly different carbohydrates. And that underlies some of their interactions, and I'll tell you about that in a little bit. One other point to make on this tree of life that we're looking at is that there are many different kinds of bacteria, and that's symbolized by these branch points. This is a vast underestimate. I mean, there are tens of thousands, if not more, types of bacteria, probably hundreds or thousands in the world. The type of bacteria that live in our own bodies, it's probably around between 5,000 and 10,000 types. Questions or comments up till now while I switch back to the PowerPoint? Okay, so the bacteria that live in or on our body are called the microbiota or the microbiome. And the definition of the microbiota or the microbiome is the microorganisms, which is mostly bacteria that live on or in larger multicellular organisms. And I say mostly bacteria because there are also fungi and viruses and so on, but this talk focuses on bacteria. So a lot of these bacteria live on our surfaces, and the human body has an outer surface, of course, which is the skin, but there are also inner surfaces of our body, which is our GI tract and the vaginal tract and the lungs, because those are parts of the body that, although there are folds to get into them, like in the mouth, for instance, of your cheeks and all the way down, and also from the bottom end up, but technically the inside of your GI tract is contiguous or attached to the outside world, and the rest of the human body is separated from it. So the whole GI tract is considered a barrier surface between the human body and the outside world. And similarly, the skin has a barrier surface, and the lung has barrier surfaces, and the vagina has barrier surfaces. And so on these barrier surfaces, there are different communities that live in each of these parts of our bodies. And believe it or not, there are as many bacterial cells that live in and on our body as there are human cells. It's considered to be about a 1.3 to 1 ratio for cells. So that means that each of us walking around sitting on this Teams call is actually a walking, talking ecosystem of not just humans, but an entire collection of organisms. And when you think about that, and you think about how many genes we encode, let's say we encode about 20,000 genes, and each bacterium, let's say, encodes about 1,000 genes, that means that the size of the genetic reservoir of information that bacteria encodes is itself 200 times the number of genes that we have. And so these bacteria encode so many genes, and they eat some of the same foods that we eat. And so their function rivals that of any organ in our body. So I know a lot of you are working in labs around Sloan Kettering, and some of you are studying the lung or the GI tract or the heart or the brain or cancers that arise from those organs, or the blood system, or maybe endocrine glands like the thyroid. But the bacteria that live in our gut and elsewhere could almost be thought of as an extra organ that produces substances that our body can respond to. This is a few other fun facts about the microbiome. A lot of the microbiome research that I do and that most people in the field do, although not everybody, focuses on the ones that live in the GI tract, more so than the other parts of the body. And the reason for that is that 99% of the bacteria on our body live in our gut. And this barrier surface between the GI tract that makes up the GI tract that separates the human body from the bacteria, if you were to uncoil it and spread it out, here it is all coiled up, our GI tract. If you were to spread it all out, it would be the size of 2 tennis courts. So we have an enormous population of bacteria on one side, a huge surface area, and then the rest of the body, including the immune system and the digestive functions. So there's an enormous amount of interaction that goes on between the parts of our body and the bacteria that live within us. And we co-evolved with these organisms. Yes. Let me give you one other fun fact about the microbiome as a way to try to convince you that it's really important, and then we'll go into some of the scientific aspects of how we study it. If you think about, so one interesting feature of the human body is that, and all mammals, is that mammals make milk that feed their young. Breast milk contains certain types of sugars that babies cannot digest. These are called human milk oligosaccharides, HMOs. And sort of have to wonder, why would a new mother who's undergone this tremendous stress of carrying a baby to term and then producing all the milk that the baby needs, why would the mother have evolved to produce substances in breast milk that the babies cannot digest? And it turns out that these human milk oligosaccharides are some of the favorite foods of certain types of bacteria, for example, bifidobacterium. And this is one of the lines of evidence that suggests that we humans have evolved to help cultivate and coax along and encourage certain organisms to grow inside their GI tracts of our babies. Okay, so traditionally, how do people study microbes? As I mentioned, looking at them under the microscope, that's one way. Another way is you can cultivate them. And Robert Koch invented a way to grow bacteria on a petri dish. Perhaps some of you have done this in your biology classes or in laboratories. And this approach is great, and 100 years of microbiology have been done in this way. But it's biased. And it's biased in that the bacteria that grow are the ones that can grow under the conditions that you set up for them. The temperature you choose, the amount of oxygen in the environment, what type of food or sugar you put into this plate. So that when you do that, specific bacteria will grow under these conditions and other ones won't. And the limitation of this approach is that you only identify a fraction of all the microbes. About 20 years ago, a professor named Carl Woese invented a new way to characterize the bacteria that were in a particular sample that did not involve culturing them on a petri dish, as pictured here. And that approach relies on the idea that there's a gene that all bacteria share, and that gene is called 16S. But not every portion of that gene is exactly the same across all bacteria. There are some portions of it that are the same and are conserved and are identical across all bacteria, and other portions of it are very unique to each bacterium because over evolution, point mutations accumulate over time. And so by using PCR, polymerase chain reaction, which I suspect some of you are using in your bench experiments this summer, and DNA sequencing, which I suspect some of you are using in your labs, Carl Woese invented this approach called 16S sequencing that allows us to use DNA sequencing to basically do a catalog or a census or an inventory of all the bacteria in a particular sample using this PCR plus sequencing to then identify which bacterium is in the sample. And this approach is completely free from the biases that the culturing introduces. And when people started using this approach of 16S sequencing, an entire world was opened up, and scientists started realizing that there's a whole world of bacteria that live in the GI tract that were previously underappreciated. And the reason for that is that most of the bacteria that live in the GI tract are anaerobes, anaerobic, meaning they do not breathe oxygen. They don't respire. Instead they ferment, and fermentation does not require oxygen. And in fact, most of these are strict anaerobes, which means that if they're exposed to oxygen, such as there is in the air, they die. And so 100 years of microbiology that relied on petri dishes in incubators without controlling the oxygen concentration in the air overlooked this entire universe of life forms that are living in our own GI tracts that we're walking around with. And the moment people started doing the sequencing that was free of these biases, we realized, hey, actually these bacteria that we can culture in petri dishes, they only make up a tiny percentage of what's in our GI tracts. So that's sort of been an exciting frame shift or paradigm shift in how people look at the bacteria that live in and on our bodies. Questions or comments up to this point? Do I still have you guys? I still see a lot of faces. When I'm showing screens, I can't on my screen. I don't see your faces, but seems like people are here. Is anybody chatting anything? Please feel free to put in the chat.
But
too.
They may be unfamiliar with Teams because we typically do Zoom. But if you do have questions, feel free to type them in the chat, or you can unmute as well.
Yeah, please do.
And you're welcome. This is meant to be interactive with Tsoni.
I have a feeling, Vicky, that our students are probably better than you and me at using Teams.
Right. True.
So how do we study the gut microbiome? This part gets a little icky. Okay, so trigger warning for those of you who need trigger warnings. It's kind of yucky, but this is what we do. We ask patients or our mice in the lab to poop into a container. Well, they poop into a larger container and they get sampled into a smaller one. And then under a hood that sucks up the air so we don't have to smell it, we take little bits of it and put it in individual test tubes. These are the same test tubes that you guys are using in your experiments. And we freeze them in a biobank at -80. And then we extract and purify DNA. I imagine some of you are extracting and purifying DNA from mammalian cells in the labs that you're working with, or cancer cells. And then, as I mentioned, using this 16S sequencing, we generate the DNA sequencing data and then analyze them. So here, this is a tiny snippet of what some of that looks like. Here's a bit of a phylogenetic tree that shows which bacteria are related to the others, just like that Evogeneo life tree that I showed you guys.
Tsoni, they typed it the question in the chat. So it says, how does a C-section affect someone's microbiome / health long term?
Yeah, that's a terrific question. There is a researcher at NYU, although she's now moved to Rutgers, Maria Dominguez Bello, who has really been a pioneer of this area of research. And she has discovered and described that when a baby is born naturally through the birth canal, a vaginal birth, the baby acquires bacteria that typically live in the female genital tract. On the other hand, when a baby is born by C-section in the operating room, the baby does not get exposed to those bacteria. And usually the bacteria that those babies are exposed to are typical skin bacteria. that they probably acquire, probably not during the C-section itself because everything's sterilized in the operating room, but from their caregivers' hands and cheeks and chests as we hold our babies. And we don't know for sure because no one's ever randomized babies to C-section versus vaginal birth. But Dr. Dominguez-Bello and others believe and have some evidence for the idea that it's not ideal for babies to acquire the skin flora, the skin microbiota, as opposed to the vaginal microbiota. So she actually ran a clinical trial where she took gauze and soaked it in the vaginal secretions of mothers who were going for C-sections, and then basically wiped that gauze on the newborn babies as they were coming out of the C-sections. And she showed that by doing that, she could change the trajectory of the microbiome development of those babies to look more like natural birth babies than C-section babies, meaning the baby's intestinal poop microbiomes look the same. Let me skip ahead to one interesting slide. I see the other question. We're going to get to it in a second. There's this concept of ecological succession. Out in nature, there's this idea that there are certain organisms. If you have, suppose, an environment that just has bare rock and no plants living on it at all, certain organisms that are very hardy and require very few nutrients. And the example in this case are lichens. And when they form, they are able to grow under extreme conditions with very few nutrients and environments that are not easy for other organisms to grow in. And when they die and they decay, they then become food sources and provide a nice environmental niche for other things like small annual plants. And then eventually you can have from these pioneer species grow intermediate species, and finally you arrive at what's named the climax community, which has much larger, much more complicated organisms that have much more sophisticated needs for growth. This is called primary succession. And there's a metaphor that this might be what's happening in the GI tract of a newborn baby, that initially there are some organisms, specifically ones that come from the female genital tract, that can grow optimally under the conditions of human milk oligosaccharides in the breast milk and live on their own. And then later, as the baby matures, particularly when the baby weans from breast milk to regular food and is exposed to all sorts of other bacteria and nutrients through the food, and also babies are constantly putting everything in their mouths, then gradually you have a more and more mature community. And people who study the GI tract microbiomes of babies have shown similar things. The other half of the slide is that there's something called secondary succession. And this is what relates to cancer therapy. I know a lot of you are involved in this program because you're interested in human health and cancer. When a forest undergoes a forest fire, it wipes out almost everything. And then again, you might have a year later, pioneer species forming, intermediate species, and climax community. And if we have time, well, I don't know if we do, but in the context of patients who get treated for cancer, sometimes something very similar to these forest fires happen. And that's one of the things that I study in my laboratory. When we give patients massive doses of antibiotics and their nutrition gets really crummy because they're feeling very sick and they're getting cancer treatment, sometimes we wipe out or at least injure their gut microbiome. And then they have to go through this process of rebuilding their own microbiomes. And an analogy for that is here, this acronym HSCT, which is hematopoietic stem cell transplantation, which is the type of cancer treatment that I give to the patients I take care of. So there are these interesting metaphors or analogies between how a ecology community of trees and plants have similar patterns to what happens in a newborn baby and has similar patterns to what happens in the context of cancer treatment. There's another question. When did next-gen sequencing come into play, and how long have we known about these anaerobic bacteria? From Robin. Great question. So Carl Woese did his work in the 1980s and 90s. I mean, PCR was, anyway. So early on, there was not next-generation sequencing, and people used old-fashioned Sanger sequencing with these 16S PCR amplicons. Next-generation sequencing really sort of came around in the aughts. And it's really in the last 15 years or so that microbiome research has really taken off. And you can sort of get a sense of that if you type in microbiome into PubMed, there are these little, it shows you a little graph of how here, of when people really started studying something. So this field really took off, you know, around 2012, 2014. And at this point, you know, in 25,000 articles. All right, so I mentioned that we collect all these microbiome specimens at MSK, and we have a core facility with a bunch of freezers, and we've collected, you know, many thousands of samples from thousands of patients, and we're using that to study what happens to the gut microbiome of patients who are undergoing cancer treatment. And I'll tell you, without getting into the details, because we're limited by time, but I would love to talk about this more with you, anybody who's interested.
Since we started a bit late, it's okay. Don't feel rushed, Tsoni. Go ahead. It's okay.
A few minutes later. All right.
Yeah, it's okay.
Thank you, Vicky. The gut microbiome probably has an influence on many aspects of our life, of our health, and I'll go into some of that in a moment. So I'm going to skip some of these things. I don't have a slide on this, so I'll just tell you, and then I'll jump ahead to what happens in cancer treatment. So it probably influences our metabolism in terms of our nutrition, because a lot of the food we eat, we cannot digest ourselves, even adults, never mind the human milk oligosaccharide business I mentioned earlier. But some of the foods we eat, we cannot digest fully without our gut microbiomes. So it affects our nutrition, because then the bacteria ferment the food and break it down into molecules that we then can absorb and use as energy sources. So the microbiota modulate our nutrition. They also modulate our immune system, because our immune system has to recognize our self versus foreign things, but our immune system also has to be tolerant and to accept and be in harmony with the bacteria that live inside us. And so it also affects cognition and emotions. People think those connections are more tenuous in the literature, but people have found that as well. Okay, so let me tell you a little bit more about these injuries that happen to cancer patients while they're getting treated for cancer. Are there other questions? If anybody wants to pop in the chat as I transition to that next topic. Okay.
Did you answer the second question in the chat from Robin?
Yes, about
okay, great.
I didn't explicitly ask how long have we known about the anaerobic bacteria. I mean, I don't know when the first anaerobic bacteria were discovered. I can't answer that. That would require a Google search we could do together if you'd like, Robin. But I can tell you that it was only in the 90s that it was first identified that there are more bacteria that are anaerobic than aerobic that live in our guts. Okay, so one thing that we measure when we analyze a bunch of bacteria in a community sample is we ask about how diverse is the sample. I know that diversity is a hot topic in society right now, as it should be, and we all agree that our society should be more diverse, and we should celebrate diversity of different human phenotypes, like people who come from different ethnic backgrounds or different genders and so on. That's a sort of a social construct of what diversity is. Biologically, how would you measure diversity? So basically what you do, I want to show you guys this one slide. Let me pop it up because I think it's an important concept. I thought I put this slide in here, but it is not. I just want to get it 1 second. I will find it. I'll take 2 seconds. So when we think about diversity, we measure 2 different things. We measure how many different organisms are present, and we also measure how evenly they're distributed. So again, using the forest as an analogy, here's a forest community, and there are 4 types of trees, A, B, C, and D. So this the simple count of how many different unique species are present, the technical term for that is species richness. So this ecosystem has a species richness of 4. And in this case, they're evenly distributed. There's 25% of in this community of each of these 4 trees. Now imagine another community that has the same richness, 4 types of trees, but 80% of them are all type A, and then there's only 5 or 10% of type B, C, and D. So this idea is called evenness. Here there's a lot of evenness, and this one is not very evenly distributed because there's an imbalance in the species count. So there are these mathematical diversity metrics that take into account both richness and evenness. And that's what we're measuring in this graph that I want to walk you guys through.
Tsoni, I wanted to highlight what Carlos wrote in the chat. He said, isn't there some sort of index and like species richness versus species evenness?
Yes, there are. There are indices that specifically measure one versus the other. And what you mentioned about Simpsons is exactly the one that I'm using here in this plot. It's a great question. But okay, this one's called Shannon, but there's a Shannon diversity index. There's a Simpsons diversity index. You're absolutely right. So whenever we have a sample that we've sequenced and we've counted how many organisms are present and how many of which type of organism, we can use these mathematical formula to calculate a diversity value. And the higher the value, the more diverse the sample is. And that takes into account, as we mentioned, both evenness and richness. So what this plot is, each dot is a stool sample that was taken from a patient that got a bone marrow transplant here at Sloan Kettering. When we give patients bone marrow transplants, they usually have blood cancer, leukemia, or lymphoma. And that leukemia or lymphoma is so bad and so life-threatening that we have to give the patient doses of chemotherapy that are so toxic that they might kill the patient, because they would wipe out not only the leukemia, but also the normal blood system of that patient, which resides inside the bone marrow. And so what we do is we wipe out the patient's entire blood system with mega doses of chemotherapy, sometimes also radiation, and then we give back to the patient the blood system of a donor by removing bone marrow cells from a healthy donor and transplanting those cells into the patient. And so that's on the time scale of transplant, that's what we call day 0, the day of the transplant. So first we give a few days, like a week or 2 of chemotherapy and radiation. Then the patient gets their cells. Then there's a period of time that it takes for the cells from the donor to set up shop in the patient's bone marrow and start producing new white blood cells, or new blood cells of all sorts. So what we did is we collected stool samples from a bunch of different patients. And you can see that there's a big spread. Some of them are very diverse. Some of them come in with very low diversity. But here, if we draw an average line that takes a sliding window, and in each window calculates the average of all these points, and then we plot that average as this gray line with an error bar around it, or a confidence interval in the light gray, we can see that there's a dramatic trend of downward diversity during the time of bone marrow transplantation. And this has been observed over and over and over again in many, many populations of transplant patients. I like to call this a microbiome injury. There's a loss of diversity of the gut microbiome. And here's another example of this, just in one single patient. So this patient had their transplant here on day 0. The blood count of their white blood cells was normal. We gave them the transplant. It drops down to basically 0. That's with the red dots. And then the patient recovers and their white blood cell counts come up. Here's the chemotherapy we gave them. And the patients also often get a bunch of different antibiotics because they get fevers and they get infections during this time. And each of these bars is a poop sample that goes from 0 to 1, where 1 or 100% represents all the different organisms. And in these stacked bar plots, each level represents a certain bacterium. So this is one microbiome sample, and the fact that it has many different colors here represents that each color is a different bacterium, and so it's a normal, diverse, healthy microbiome. And then around day 0 of this transplant, suddenly we see an expansion of one organism that ordinarily is not present in a patient's microbiome. In this case, it's called Enterococcus faecium. And this Enterococcus faecium expands to take over the entire gut microbiome. So the whole poop sample only has one type of bacterium in it instead of a few hundred. And this sort of domination state is a really common pattern in bone marrow transplant patients, but it's not seen almost outside of bone marrow transplantation. So like none of us on this call have 100% of our GI tract being filled with just one bacterium. And I'll tell you, this one type of bacterium can also cause bloodstream infections. And we've also demonstrated in some of our research that it can cause or at least exacerbate other problems that transplant patients have. For example, graft-versus-host disease, where the donor's immune system attacks the patient. So one of the things that I study in my laboratory is what are the conditions that set up this microbiome injury? What are the consequences of it to the patient, and what could we do to prevent it or treat it? Okay, so what could we do? In the last few minutes, let me shift to what can we do to the microbiome? Well, people always ask about probiotics, and that's a great question. This is another phylogenetic tree like the one I showed you from the Evogenio website. Each tip is a different type of bacterium. And we've color-coded in blue the bacteria that are good for bone marrow transplant patients based on our research. And in red are the ones that seem to be associated with bad outcomes for our patients. And by the way, these blue ones tend to be strict anaerobes here. They're anaerobics. They need low oxygen environment. And a lot of these potential pathogens that cause problems are typically aerobic. Now in green, my colleague Ying Tower in the infectious disease department has marked here all the different bacteria that you can find in probiotics that you can buy at CVS or at GNC or on the internet. So it kind of gives you a sense that at least for bone marrow transplant patients, maybe we shouldn't give them the kind of probiotics that we can buy on Amazon. We'd like to try to develop probiotics. And in fact, we have clinical trials where we're doing that, where we and our collaborators at companies that do this have hand-picked certain bacteria from these blue groups of the good bacteria. I put good in quotes because the bacteria aren't good or bad. They're just, we view them as good because they're helpful for patients. And we're trying to see what happens if we give bacteria next generation probiotics, if we give patients next generation probiotics that are comprised of these so-called good guys, if you allow me to anthropomorphize. So that's a comment about probiotics. There are a couple of other approaches that I'll mention to you, and then I'll stop for a few more questions. So one approach to probiotics is to give a pill that has live bacteria in it. And another approach that I just mentioned might be to try to be smarter and more clever about which strains we put, which bacteria we put into these pills. Another approach, again, trigger warning for the ick factor, is a fecal transplant. This is actually a very old idea that has been practiced for thousands of years, where you take the poop of someone who's healthy and you give it to a patient who's sick. There's a certain type of diarrhea called C. diff, Clostridium difficile diarrhea. And C. diff causes problems for people when the normal gut microbiome is messed up. That opens up an opportunity for C. difficile to grow more. And although there are some antibiotics that can treat C. diff infections, one of the ways that C. diff is treated nowadays is with a poop transplant from a healthy person. Now, you can't possibly ask a patient to eat someone else's poop. I mean, that's ridiculous. So how do we do that? One way is you can do a nasogastric tube where you place a tube in someone's nose and it goes down their esophagus and opens up in their stomach. And then the nurse or the doctor can just inject something into the tube and the patient doesn't have to actually swallow it. That's an old-fashioned way to do it. It's still done. Another way is also through a tube, but from below. That's sometimes called a colonoscopy, where a gastroenterologist, a GI doctor, goes in the butt with a camera and a tube, and then you can also inject things through the tube. And then a third way to give a fecal microbiome transplant is to package the poop in pills that don't smell or taste like anything. And then that's done in a very clean way, and the patients can swallow the pills like any other. Now that all seems awfully disgusting, and you might think I would never ever want to do that. But let me tell you, C. difficile diarrhea can be very troubling, and people can suffer a lot from it. And believe me, there are patients who have such bad diarrhea for so many months that they get to a point where they're willing to try anything. And it really does work for C. difficile. And there are a lot of clinical trials that are asking, well, what else can we use fecal microbiome transplants for? Can we use it for cancer? Can we use it for inflammatory bowel disease like Crohn's or ulcerative colitis? Can we use it for other problems? Anyhow, those are all different approaches to what we call probiotic approaches to try to treat the gut microbiome or fix it. And there are a few other approaches. I'm just trying to see if there's anything else in the chat. How do you rebuild a microbiome? Okay, how do you, so Amy, I just answered one way to try to rebuild a microbiome after transplant. Another question is stem cell transplant the only way that loss of diversity could be remedied? Okay, so Emma, you're asking here kind of two different questions because I'm talking about two different types of transplantations. One is a bone marrow transplant where we replace the blood system of a patient. The other is a fecal poop transplant where we give a patient a donor's poop. We've actually done a clinical trial where we did both. Patients were getting bone marrow transplant and they also got a fecal transplant. So we don't really use, the reason we do the stem cell transplant is to treat leukemia, and the reason that we might do a fecal transplant is to fix diversity of the gut microbiome. When you perform BMTs, asks Kevin, how do you determine which bone marrow donors will work with the patient's immune system, and how likely is graft-versus-host disease during these bone marrow transplants? That's a great question. Matching a bone marrow transplant donor to a recipient is a whole field in and of itself, and there's a protein, a family of proteins called the major histocompatibility complex, MHC, or in humans it's also called the human leukocyte antigen, HLA. And this is a group of genes that is really polymorphic. It has a lot of diversity in it across the human population. And so the approach is that we do DNA sequencing of potential donors and of the recipient, and we try to pair up a donor and a recipient who have the same HLA genes or the same MHC genes so that they'll be compatible with one another. And graft-versus-host disease, which is when the donor's cells recognize the recipient as foreign, even though we try to match as best as we can, that happens in about half of the patients, even with all the best technology. Most of the time, the GVHD is rather mild, and you can treat it with steroids and it goes away. In a minority of cases, but a sizable minority, the GVHD does not respond to steroids, and then it can become a real problem for the patient, and in some cases even kill the patient. And that's particularly tragic because many times we're able to completely cure someone's leukemia with a bone marrow transplant, but then they end up getting graft-versus-host disease and they die of that, which is so devastating because they actually finally beat the leukemia that was their original problem, but then they die of the side effects of the cancer treatment. And so that's one of the things that actually motivates my research and my work with patients is it's not just can we find a cure. Like, we have cures. We could cure cancer, we could give everybody massive doses of chemotherapy, not every cancer, but many cancers, we can cure them with either massive doses of chemotherapy or transplant. But sometimes the toxicities, the side effects of the cancer treatment are so great that you'd end up killing the patient anyway. And so there's sort of two, a yin and a yang of cancer research. One is to understand what makes the cancer cell tick and how can you target it. And that's critical. And I know a lot of you are working on that in the labs that you're embedded in this summer. But a whole other half of cancer research is figuring out ways to make the treatments we already have less toxic and easier to handle for the patients. And that can be just as important, because if you can make a treatment like bone marrow transplant easier to handle and less, and more safe, then you could give that treatment to more and more types of patients. Is it possible that a donor might never match with anyone? Yes, it is possible. And in those cases, sometimes we can't do a transplant at all. There are ways to now do a bone marrow transplant nowadays with a graft, meaning the donor cells that are not a complete match. One of those ways is called cord blood transplantation. Usually we take the donor cells from the donor, either from their bone marrow directly with needles, or we give the donor drugs that push the stem cells out into the bloodstream, and we collect it by drawing blood. But you can also get the transplant material from the placenta and through the umbilical cord, and that's basically the baby's cells. And for reasons that we don't really understand very well, baby immune cells can be more tolerant of mismatches than adult cells. And so cord blood transplantation is one way that you could theoretically get a transplant to anybody who would want one. And another way is there are approaches to use family members who are not a perfect match, but are only a half match. Like, let's say you share half of your genes with your sibling. So maybe your sibling could be your donor if you needed one and you couldn't find a fully matched donor out in the databases.
Only I'm not sure if you saw Anthony's question from earlier. They were asking, do you have a statistical percentage for the risk rate of that intensity of chemotherapy?
Anthony, the risk of what? Of which outcome?
I think he's typing in the chat.
Sure. So I think you were asking about the risks of having a microbiome domination. Yeah. So about a third of our patients get enterococcus dominated. This was a survey that we did of microbiome injury in bone marrow transplant. Figures. Where are the figures? Here. Okay. So this is what's called the cumulative incidence of domination. And we studied this in 4 different transplant centers. So as time goes on, this is hematopoietic cell transplant day. Day 0 is the day of the cells. So as time goes on, anytime a patient gets a domination event, the curve goes up by a little bit. And what we saw is that by the time the patients are like 2 weeks into their transplant, something like 80% of them have had a domination by some organism. This is the incidence, and then the prevalence means at any given time point what fraction of the samples or the fraction of the patients have it, and it goes up to about 80%, and then as they recover it improves. And then here we're looking at what are the organisms that cause these domination events or that end up being dominated, and we have this color-coded chart of all the different bacteria that we find. Here's in green is that enterococcus I mentioned earlier, and you can see that The most common one is Enterococcus, and it's seen in about a quarter of the patients. But also other organisms that can do it are things like Streptococcus in this pea green, or Lactobacillus in blue. Sometimes in yellow, staph can do it. And then, by the way, having these domination events raises the patient's risk of having a bloodstream infection with the same organism by between 5 and 50-fold, depending on the details and the type of organism. Okay. Other questions or thoughts? I'll leave you guys with a very last slide, or I'll just go back to the other slide I was looking at, which is this one about other approaches. So probiotics is one approach, and we talked a lot about poop transplants and so on. There are other approaches too. I'll just mention them to whet your appetites for further reading in the future. I mentioned that the bacteria eat the same foods that we eat. We're feeding them. So there's an idea that if you could figure out which are the right bacteria whose growth you wanted to promote or whose growth you wanted to prevent, maybe you could eat a certain type of food or avoid a certain type of food that could help cultivate or sculpt the bacteria that live in our guts. And the cute name for this is prebiotics, because you're manipulating something that's upstream of the probiotics. And I also mentioned that the bacteria, as they digest the food that we feed them, they ferment it, and then they produce metabolites, which are their waste products. But some of those are viewed by us as yummy food, or not yummy food, but nutritious food that we absorb in our GI tract. And so maybe if we could figure out what the right metabolites are that bacteria are making that modulate our bodies in a certain desired way, perhaps we could just bottle up those metabolites and give those directly to the patient without having to deal with giving patients bacteria. That approach is called a postbiotic approach. And then another one that's kind of simple-minded but still really important is to be more thoughtful about our use of antibiotics and to avoid them when we don't really need them. For instance, if you have symptoms of an upper respiratory infection, the sniffles and a cough, and let's say it's COVID negative, and it goes on for a few days and you're really miserable, should you take antibiotics? Well, if it's a viral syndrome, you probably shouldn't, because you're going to be damaging your gut microbiome for something that's a viral infection and not a bacterial infection up here in your nose. And maybe we can be more thoughtful about which antibiotics we give cancer patients and when we give them and how long we give them for. So these are all the approaches that I'm hoping to develop in my research laboratory, and that many others of my colleagues in the microbiome field are working on to try to target the gut microbiome to improve human health. Questions and thoughts?
Thank you so much, Tsoni. And I did want to highlight, not specifically with your science, but since we do have a target audience of students, could you share a bit more briefly regarding your trajectory? I know I read your bio, but it seems as though your interest in STEM was early. You did programs in high school and so forth. So what advice could you provide the students?
Sure. I was always interested in biology. I just always had a fascination for it, and it helped that my mother is a biologist. However, math was not always my favorite subject, and I was also really interested in other things. I was interested in theater and music and politics. And when I went to college, I actually majored first in political science. My father was a mathematician. He told me that his opinion was that he didn't want me to just major in something that was like what he considered more soft fields, like humanities, say sociology or literature, art, political science, history. He considered all of that to be interesting and important, and he read those materials, and he loved reading about all those fields. But he said, what he told me was you ought to have at least one major that's a more hardcore topic, math, science, engineering, or a profession that you could, sustain yourself right out of college, like business or engineering or some sort of a professional trade like accounting. Something that is like not just I'm going to think about the world and try to write something and hope it gets published and, That was his attitude. Now that's a little closed-minded, and it's not in line with the liberal arts education philosophy of the United States. But I'm really glad that he pushed me to do that, because I ended up double majoring. And I really think that if you're bright enough to be in this program this summer and you're doing bench research as a high school student, you're bright enough to double major in college. We all have enough bandwidth to do that. And I would really encourage everybody on this call to try to double major in one thing that's technical and quantitative, computer-oriented, math-oriented, science, STEM-oriented, and absolutely enjoy your favorite art / literature / humanistic, fun, deep, philosophical, metaphysical topic. Because when you're 17, 18, 19, you don't know what you're going to want to do when you're 22 and 23. And you want to set yourself up to have something that you can easily get into a graduate program of your interest, or start working in the working world and have marketable skills. So my main advice to you is to maintain an open mind and consider double majoring, and not just in two fields that are closely related. That would be my main advice about career development for high school students.
Thank you so much, Tsoni. Anthony has a question in the chat as well.
After you transplant the microbiota to a patient that has a risk of the diversity being dominated, what stops it from being dominated again after the transplant? Great question. When you do a fecal microbiome transplant, there is no guarantee that the repair that you do, assuming you even repair it in the first place, will be sustained, because the GI tract community is in constant flux. It's in flux based on the food we eat. It's in flux based on fecal oral contamination. That's part of our day-to-day life that although it's disgusting to think about, it's there. And it's in flux because of our own metabolism and our diet. So there is no guarantee of that. The hope in the case of C. diff is that when you do a fecal microbiome transplant, is that you might be in kind of a rut with C. diff. And if you could just get C. diff down once and get some sort of a diverse microbiome in its place, that there'll be enough diversity that it'll be self-sustaining. But that is a completely hand-wavy explanation, and it's really poorly understood what drives a gut ecosystem to remain in a diverse state versus lose its diversity over time, if you remove the obvious environmental insults. These are also open questions, not just in the gut microbiome, but in ecology more broadly. Like, what drives a species to become extinct, or what drives the biodiversity of marine seaweeds to be high or low in a particular segment of the ocean. These are active areas of investigation. And when we do our clinical trials of these rationally designed probiotics, we honestly have no idea how to dose them. I mean, we kind of make it up. Should we give 1 day, 2 days, 10 days, 4 months of the drug? We really don't know. We start with teeny tiny trials where we make our best guess. We do mouse experiments with different durations and different dosages, and then we try to flip that over into humans, and we hope for the best. That's part of what's fun and a little bit of the art of designing experiments, is that sometimes you have no idea what dose or how long to give something, and you have to try and tinker and do you have to be willing to fail, and you have to be willing to tolerate long chains of failures before you get to something that actually works.
Great. It seems as though there are no other questions, but again, we'd like to thank you, Dr. Peled, for your lecture this afternoon, and thank you everyone, the audience, for pivoting due to technical difficulties. But I do hope you found that the talk was helpful, and we do plan to post the recording later for those to view. And we look forward to seeing you guys for our next series, which will take place on the 11th. So there will be a break next week. We won't have a lecture, but the following week on the 11th, we will have a lecture with Dr. Murray. Great. Thank you again, everyone, and have a good rest of the afternoon.